Hello students, welcome to the Cell Virtual Laboratory for Microbiology. In order to do this laboratory, you will need to go to cellsalive.com, and this should work on you do not need to have Flash Player enabled to do this activity. We will be looking at 3 different cells and comparing their anatomy with one another. We will be looking at plant cells, animal cells, and bacterial cells. Let's begin. To begin, let's go ahead and click on Plant and Animal Cells. You'll see here that it says Start the Animation. We can go ahead and click on this here. From here, we can choose Animal Cell or Plant Cell by clicking here. Let's start with the Animal Cell. This pulls up our animal cell. When you open up either your Word document or your PDF that I have attached to this laboratory assignment, you will notice the last few pages have models that need to be labeled by you for each of the cell types. I suggest opening up this document or printing it and completing the assignment while you are doing the online activity. Once you are here, you can click on each of the organelles and the system will identify the structures for you. Here, I'm just going to briefly go through each one in case for some reason you're having difficulty with the program. Remember, you can always pause this video if you need to look at something specific. Here we have the nucleus, the nucleolus, the cytosol, the centrosome, centrioles, which makes up part of the centrosome, the Golgi body or Golgi apparatus, lysosomes, peroxisomes, secretory vesicles, the cell membrane, mitochondria, the cell vacuole, and notice when I get to cell wall, there is nothing highlighted. That is because we are on animal cells, and animal cells do not have a cell wall. This will be important in answering the questions for your lab activity. When I go to chloroplast, you'll also see that nothing comes up. This is because chloroplasts only exist in plant cells and do not exist in animal cells. We can move on now. Here we have the smooth endoplasmic reticulum, the rough endoplasmic reticulum. The tiny dots here are the ribosomes, which are located on the rough endoplasmic reticulum. And here we have components of the cytoskeleton, which includes the microtubules, microfilaments, and intermediate filaments. Refer to your worksheet for an explanation of the functions of each of these organelles. Next, let's go to plant cells. Let's briefly look at the organelles inside of a plant cell. Here we have the nucleus. The darkened spot within the nucleus is the nucleolus. The liquid inside of the cell is the cytosol. Here we have the centrosome. Centrioles do not show up in this tutorial because centrioles are not shown in the plant cell, but they are located in animal cells. Here we have the Golgi body or Golgi apparatus. We have a lysosome. We have some peroxisomes. Here are the secretory vesicles. Here is the cell membrane. These are the mitochondria. And in the plant cell, you'll notice that we have a very large cell vacuole. In the animal cell, this is much smaller. Here we have the cell wall, which is present in plant cells and absent in animal cells. Here we have the chloroplasts. Chloroplasts are also only found in plant cells. They are not found in animal cells. Here we see the smooth endoplasmic reticulum, the rough endoplasmic reticulum, along with the tiny dots, which are the ribosomes, and components of the cytoskeleton, including microtubules, microfilaments, and intermediate filaments. Great. Now when you're done with this, we can go to our bacterial cell. We can get to our bacterial cell by scrolling up and clicking on the link here. This brings up our bacteria cell model. Notice with our bacterial cell, we have both gram-positive and gram-negative bacteria represented. This will be important to note the differences for your laboratory write-up. Let's look at some of the organelles of the bacterial cell. Here we have the nucleoid. Within the area of the nucleoid, we have the genophore. Here we have the plasmids, the cytoplasm. The endospore may be present, but it is not shown. We have ribosomes, storage granules, the cell envelope, which looks different in gram-positive versus gram-negative bacteria. On the outer edge of the cell envelope, we have the capsule, which also appears differently in gram-positive versus gram-negative bacteria. The smaller projections coming out of the bacteria are called pili or fimbria. The larger projections are flagella. If you click on examples, you will see examples of different gram-positive and gram-negative bacteria. I hope you enjoyed this tutorial and happy learning. And thank you for watching.